and welcome to the review on the chapter on blood. As we go through the study guide, I'll answer the questions and you'll see that a lot of this information is found within this PowerPoint, but it's not in the same places, so I'll be skipping around just a little bit. So let's do composition first. We'll come back to the functions. As far as composition is concerned, there are three parts to the blood: plasma, buffy coat, and the red blood cells, and I'll discuss these things momentarily. But here are just some basic characteristics of what we see in whole blood. If we remove some blood and spin it down, you'll see that the blood does divide into its three different parts. We have the erythrocytes, made up of just the red blood cells, which we also call the hematocrit, and I'll come back to that in a minute. Then we have the buffy coat, which is made of the leukocytes and platelets. You can see that it's a very small portion of the blood, less than 1%. And then plasma is the non-cellular part of blood. The formed elements are the cellular components, so red blood cells, white blood cells, and platelets, or erythrocytes, leukocytes, and thrombocytes are all found below the plasma because they are more dense. Now, as far as hematocrit's concerned, hematocrit is a percentage of red blood cells that you have within any volume of blood. So regardless of how much blood you take out in a sample, you'll always have the hematocrit being around 45%, plus or minus 5%. Males 47 plus or minus 5, and females 42 plus or minus 5. The buffy coat, again, is just the leukocytes and the platelets. Their numbers are greatly reduced, and we'll talk about their numbers later on. And then the plasma, again, does not have any cellular components, but it does have some proteins in it. And we'll look at that. You can pay attention to this. We're going to come back to this hematopoietic stem cell idea in just a minute. Okay, so again, we'll come back to plasma, but let's look now at what the functions of blood are. So as far as breaking it down into three different sections, you can see transportation or distribution. We move the nutrients, O2, metabolic wastes, hormones, whatever needs to be carried in the blood throughout the body, from the outside of the body to the inside of the body, and everywhere will need to be transported in blood in one way or another. You can see with regulation, there are multiple things that we regulate body temperature by taking heat carried in the blood out to the skin and having evaporative cooling take place and return that cool blood back to the core, so to speak. pH is regulated in the blood because we have buffers, and when we do the acid-base section of the course, then we'll talk in great detail about how the blood is able to buffer the pH of the blood. Because our blood communicates with the outside and the inside of us, as we take in water through drinking and eating through our digestive tract, it will go into the bloodstream and then be carried to the cells depending on what fluid needs they have, and then any extra fluid will be removed from the body through the kidneys as the blood goes out to the kidneys. And then conversely, if we have a lack of water, then our kidneys will be told to retain that water, and that water will then be put into our extracellular fluid, either the interstitial fluid or into our plasma. So our blood has a very important role in fluid regulation. And then, of course, protection. There's two parts to the protection component: that blood will prevent us from infection with the white blood cells. But then another thing I think is really neat is that blood has the ability to seal up the holes in the blood vessels that it's carried, and so that we prevent blood loss through hemostasis, which we'll go into some more. Okay, so if we go down in the study guide, you can see how is plasma a colloid is because there are a lot of substances dissolved within plasma that remain suspended within plasma, like albumin and the clotting proteins and other proteins like LDLs and HDLs that will shuttle material around, and then antibodies, of course, and the clotting proteins, like I said. So there gives an example of different kinds of proteins in your blood. Please make sure that you know albumin is the most abundant plasma protein. And as you can see there, it's made by the liver, and its job is to maintain osmotic pressure of the blood. We'll talk about more with capillary dynamics. Okay, so next in your study guide is on, and here you can see different components of the plasma. All right, and so now we want to take a look at the red blood cells. So the next question is about hematocrit. So again, percentage of red blood cells that you have within the body. Factors that raise and lower hematocrit initially have to do with the blood's ability to transport oxygen. So depending on what your oxygen needs are, your hematocrit can increase through erythropoiesis, or if you have anemia or some other nutrient imbalance or lack, like with vitamin B12, or if you have some hemorrhage, those are different ways that your hematocrit can change. Your hematocrit can also change based on whether or not your plasma volume changes. And so dehydration is going to increase your hematocrit because you have less plasma. And remember, because hematocrit is a percentage of red blood cells, if you lower the amount of plasma that you have, then you'll increase the amount of red blood cells that you have in your blood. Okay, let's go ahead and talk more. We'll come back to the hematopoiesis portion of your study guide. Let's go ahead to the red blood cell part. And so red blood cells, as you can see here, how much we typically have within our body, about 5 million per microliter, which is a lot of red blood cells that we have. And their shape, they are biconcave discs, which we'll discuss in just a minute. They are anucleate, which means they do not have a nucleus. They're very flexible for travel through capillaries, because typically capillary width is just barely larger than the width of a red blood cell, so that red blood cells at the capillary level travel one at a time in single file order. They have a huge surface area to volume ratio, which is suited for gas exchange, which I'll discuss in just a minute when we take a look at a picture of the biconcave disc. So here is what an erythrocyte looks like, top view and side view. Now you would think, why would being pinched in the middle be a better shape than if it was spherical? Wouldn't you think you could hold more hemoglobin if it were spherical than if it were this biconcave disc shape? But it's not about how much hemoglobin you can hold, but how much surface area there is for exchange. So the idea is that all along the cell membrane area right here is where hemoglobin is going to exchange oxygen, either with the tissues or in the alveoli. And the hemoglobin must come up and be next to the cell membrane in order for exchange to occur. So if this were spherical and this portion were very large in volume, the hemoglobin that would be found within the center would never have an opportunity to get to the edges, and therefore you wouldn't have an opportunity to exchange. If you've seen or read Divergent, and you know how the dauntless group of people have to jump on and off the train and the train never stops, you have to be able to get close to the edge to jump on and off. And that's exactly what hemoglobin is doing. It's getting to the edge so that the oxygen can either jump on to the hemoglobin or jump off of the hemoglobin, as the case may be. Okay, so I did the purpose. If we take a look at, here are the stages of red blood cell production. We'll come back to hemoglobin. So this is going to occur in the bone marrow, and in our bone marrow we have what are known as hematopoietic pluripotent stem cells, which have the ability to become any of the types of blood that we have, blood cells, not just the red blood cells, but the white blood cells and the platelets as well. In order for hematopoiesis, or specifically erythropoiesis, to occur, you need iron, vitamin B12, and folic acid. And here are the different stages that the red blood cell will go through. You have a question that says, what would be some situations where you might find more than normal number of reticulocytes in your blood? And this is going to be in situations where the cells can't mature properly into erythrocyte, either because we don't have enough iron or that we don't have enough vitamin B12 that we take in our diet but is absorbed by the production of intrinsic factor from our stomach. So if we don't have intrinsic factor, then the vitamin B12 won't be absorbed, and then we won't be able to finish maturation into this typical erythrocyte shape. And remember, shape determines function. So if you aren't shaped like this, then you can't carry oxygen like you should. So those are situations where you might find more reticulocytes. So lack of iron and lack of vitamin B12. Okay, so let's go back to hemoglobin and talk about its characteristics. And we'll get into CO2 transport in just a minute. Okay, so hemoglobin is a protein. It's an actually tertiary level protein. So there are four protein subunits, globin molecules that have 4 heme groups attached to them. We'll look at what that looks like in just a minute. Every hemoglobin can carry 4 oxygens because the oxygen will bind to these heme molecules right here. This is probably an important number to remember. What is the concentration of hemoglobin in our body? Should be between 12 and 18 grams per deciliter of blood. And that determines the oxygen carrying capacity of the blood. So if we take a look at hemoglobin here, you can see we have 2 beta globin chains, we have 2 alpha chains, and each chain has its own heme molecule in the middle, and here's a picture of a heme, and in the center of a heme will be an iron atom, and it's this ferric, well, I'm going to get it backwards, this form of iron has the ability to bind to oxygen. And if you wonder, well, why is blood red? Well, it's because iron oxide is formed right here, and that's a red color, and so yeah, your blood essentially is rusty. Okay, so there is that. Why is oxygenated blood brighter red and deoxygenated blood darker? It just depends on how many hemoglobin molecules are fully saturated with O2. After blood has been out to the tissues, typically the hemoglobin returns back to the lungs only 75% saturated, and that will result in the darker red color. Okay, what are the changes hemoglobin goes through as a person grows from an embryo to an adult? Fetal hemoglobin has a much greater affinity for O2 than maternal hemoglobin, and justifiably so, because a fetus has not the exposure to oxygen like an adult does, and so whatever oxygen is presented or is available to the fetus, then that hemoglobin has to bind much more readily, and so that's what happens, is that fetal hemoglobin has a greater affinity for oxygen than adult hemoglobin. Okay, I'm going to make sure that I haven't missed anything. I don't think so. Okay. Here's just a little bit about, now we'll go into more of the erythropoiesis in 2.1.6. So what is erythropoiesis? That's the process of producing red blood cells. And what signals the production of red blood cells will be erythropoietin from the kidneys. Why would the kidneys produce erythropoietin? The epithelial cells within the kidneys will detect the hypoxia, which means the low oxygen levels within the tissues. And when the epithelial cells of the kidney are hypoxic, then that erythropoietin will be produced by them, and then that will go to the bone marrow and stimulate the hematopoietic stem cells to go through erythropoiesis. Okay, so that's when and where. Just a little side note where it says in lab you'll be learning more about erythropoietin or EPO. The reason why it's illegal in Olympic competitions is that it gives you more red blood cells, and so that oxygen will give the the athlete's muscles just that much more ATP, or allow them to make that much more ATP, and then in turn give them that edge. Now you'd think, well, what's a few more red blood cells going to do for you? Well, remember at the Olympic level, at world-class competitions where we're really testing for EPO, we're looking at tens to hundredths of a second difference in performance. And if one person has, a hundredth of a second more ATP muscle contraction causing ability than somebody else, they win the gold, and the other person wins the silver. And so that's why we don't want to give anybody an unfair advantage in competition. Key nutrients important for blood production, again, iron and vitamin B12. Here's anemia. So explain anemia and some of its common causes. Why would anemia cause a person to become lightheaded? Because you don't have enough oxygen going to your brain, and whenever that happens, then your brain lets you know by feeling lightheaded that you need to lay down, that you need to get horizontal, and so that the blood can get to your brain. Anytime your brain does not have enough oxygen, it's going to let you know by getting more blood to it by making you pass out or making you feel lightheaded. Dangers of becoming severely anemic, of course, will be inability to carry out cellular functions and ultimately death. So here are some types of anemia. So we have hemorrhagic anemia, where you're bleeding or excess red blood cell production or destruction. Sorry. In that case, you may be exposed to radiation. You may be, like X-ray or radiation therapy for cancer, or you may have malaria. The malarial parasite will cause red blood cell destruction. So that's one way to be hypoxic or have anemia. Reduced availability of O2, high altitudes or pneumonia may decrease your ability to deliver oxygen to your cells because you don't have oxygen in the first place. And then another hypoxia is if you are increasing your tissue demands for cellular respiration reactants like glucose and oxygen, then you don't have enough red blood cells to take care of the demand. You can take a look at this. This is just what the little negative feedback mechanism going on to restore normal blood oxygen levels. And then here is how we get rid of the red blood cells. So we'll come back to that because we do have a section on breaking down red blood cells. So I'll come back to that in a minute. But here is some more of anemia. So we've got 6 different kinds of anemia. So like I said, talked about hemorrhagic anemia, losing blood, hemolytic, your red blood cells lyse either because you have abnormal hemoglobin. And I put abnormal hemoglobin in the other group too. If you have thalassemia or sickle cell anemia, genetic disorders that result in a malformation of hemoglobin. Transfusion mismatch, if you don't get the right kind of blood, then your immune system is going to destroy those red blood cells. And then, like I mentioned before, parasitic and bacterial infections. Aplastic anemia, I already mentioned that, but not specifically. This is if we're destroying the bone marrow. So certain toxins that we're exposed to, like chemotherapy. We know that chemotherapy is a treatment, but chemotherapy is really toxins destroying our rapidly dividing cells, and unfortunately. But so that is usually an effect of being on chemotherapy, is that your bone marrow will not produce any of the blood cells that you need. Drugs, other kind of drugs besides chemotherapy can affect that. And then here's the ionizing radiation if you're taking radiation therapy for cancer as well. If you don't have enough hemoglobin, that's also going to result in anemia. So iron deficiency anemia, we get the microcytes, we get reticulocytes being produced. Pernicious anemia, lack of vitamin B12 along the way in producing adequate structure of the erythrocytes. and again, the abnormal hemoglobin, either genetically because you have sickle cell anemia or thalassemia. Here's a good graphic that shows what a normal red blood cell looks like if you have normal hemoglobin. So you can see here in this 6th amino acid, glutamine is the amino acid that's supposed to be there out of the 146 that make up hemoglobin. But you get one base pair substitution in the gene that will result in valine in place of the glutamine, and so what that does is cause the beta chain to have an abnormal shape, and again, structure determines function, so if you don't have that right shape, it's going to affect the function, and this is what happens to the red blood cell. In being sickle shaped, two problems arise. One, you can't carry oxygen like you should, and the second thing is you're going to get stuck in the capillaries that are out in the tissues, and we can get organ damage and organ failure as a result of sickle cell anemia. On the other hand, if you have too many red blood cells, then it's going to make the blood very viscous and sludgy, like in blood doping, either with takeout blood and put it back just before your competition like we used to do until we could make artificial EPO or synthetic EPO. But now we got the EPO, and that's going to give you polycythemia too. So that's going to increase your resistance to flow, and we'll talk about that when we get into the blood pressure regulation section of this. Okay, so I think I've gone through, we got to go back to the breakdown. So here's what happens when we break down our red blood cells is that aged and damaged red blood cells will be engulfed by macrophages of the spleen, liver, and bone marrow. Hemoglobin is broken down into the heme and the globin. The globin then is broken down to amino acids, which will just go right back into your bloodstream that we can use for whatever other proteins that we want to make. The heme then is broken apart. The iron is going to be ferried by transferrin or ferritin or hemosiderin, and stored within the liver primarily, and then bilirubin is the leftover part of the heme molecule, and let's follow this pathway. I didn't finish this one. So iron is bound to transfer and released to the blood from the liver as needed for erythropoiesis, and it'll go back to the bone marrow, and then we can make new red blood cells again. So here is the bilirubin, and then we've got two kinds. We've conjugated and unconjugated bilirubin. The conjugated kind is going to go into the bile and then secreted into the intestine in the bile, where it's metabolized to stercobilin by the bacteria and then excreted in the feces, or we can reabsorb the bile salts that'll be in there, and then that'll go back up in here and back to circulation. So there's the breakdown of the red blood cells. The question says, what are some of the causes of jaundice? Jaundice is your liver's inability to process this bilirubin, and so instead of putting it in the bile, then it's going to stay out in your bloodstream, giving you that yellow color. You'll see it in your skin. You'll see it if you have a darkly pigmented individual, palms of their hands, and soles of their feet, or in the whites of their eyes and the sclera of their eyes. That's how we can see that. And so we have to, typically if babies, expose them to ultraviolet light so that that can be broken down, and kind of looks, it says, what does it look like? Well, kind of looks like, when you bruise and it turns that yellowy color, that's basically the bilirubin. And then as far as causes, anything that can be wrong with your liver. If you have hepatitis, any one of the hepatitises, A, B, or C, if you have cirrhosis of the liver due to liver damage from alcohol consumption, or, I just had it and I just lost my thought. Hepatitis, cirrhosis, cancer, liver cancer. So anything that can affect your liver then will result in jaundice. Okay, and then lastly, white blood cells and platelets. So let's go on through here and give you some basic information about white blood cells and platelets. we'll have a lot more information when we get to the immune system as far as the white blood cells are concerned, and then in the hemostasis stuff, which I'm going to do now too, we'll talk more about platelets. Okay, so as far as the leukocytes are concerned, this is how many we have, 4,000 to 10,800 per microliter. You know, in a red blood cell, we have 5 million per microliter. So many, many times more red blood cells than we have white blood cells. What's their job? Defend against disease. Now, disease, we almost never have disease in our bloodstream. We're always striving to make sure that we don't have infection within our bloodstream, and therefore we keep the infection out there in the tissues. Well, if the white blood cells are the defenders and we don't ever get germs inside of our bloodstream, then what's the reason for having them? Well, they have to have the ability to be carried throughout the body, and then they have to leave the bloodstream and go out into the tissues and do their defending out there, specifically in the lymph nodes, but out in the tissues. So there's a process where the leukocytes leave the blood called leukocyte extravasation. So extra means outside of vasation, the blood vessels. So there are three things that have to happen. First of all is margination. The capillary epithelium will express, or endothelium will express some molecules called cell adhesion molecules by chemokines released from the injured tissue and from the resident macrophages that are out in the tissue. And when these chemicals act on the capillaries, they put little hooks basically on the inside of the capillary, and then on the surface of the white blood cells, on the phagocytes, primarily the neutrophils and macrophages, they too will express hooks on the other side. So it's really like Velcro, actually like super strong Velcro. So as the white blood cells roll along on the endothelium anyway as it's moving through the blood, excuse me, so when these cell adhesion molecules are expressed, then the white blood cells, the phagocytes will stick to the capillary wall, and then a change in shape, and I'll try to post a video link for that. I will post a video link. It's kind of cool how it happens. So then they'll change their shape, their cellular conformation, and then they'll go through diapedesis, where they will slip through where the endothelial cells meet each other in the capillary wall, and then chemotaxis will happen, where they will travel, you know, toward where the infection is, and then they move by amoeboid motion. Anytime that we have an leukocyte number greater than 11,000, then leukocytosis has happened, and we just know that you have some infection. Okay, so this just shows the different leukocytes, and since that's not in here, and I'm guessing that it's going to be in with the immunity section, then I'm not even going to talk about that. So we'll just skip all of this, and you can look at it if you want. All right, so here are the platelets. So the platelets come from, again, these hemocytoblasts that'll be in the bone marrow, and they start out from megakaryoplasts, become a megakaryocyte, and then they'll fragment and become platelets. And so platelets are not actual cells, but just fragments of these megakaryocytes. And then their role, of course, is in hemostasis. So I'm going to stop this video here and then go to and start specifically with hemostasis as the next video.